Okay, hey people. So this is going to be regulation part 1. So instead of cutting out our last day of metabolic regulation, we're just going to kind of fuse them together. I couldn't figure out how to just ditch one of the days. They go together and they don't really make too much sense without each other. So there's going to be a 2-part video. The first one is going to describe all the ways that enzyme activity can be regulated in a cell. And then the second part is going to go into specific examples of how some of these methods are used specifically in the metabolic pathways. So we're going to go in this order. So you should be able to describe how each of the following regulates enzyme activity: pH and temperature, Zymogens, Pest and N-terminal sequence identity, transcriptional regulation, post-translational modification, substrate product concentrations, and inhibitors and activators concentrations. Okay, this is a figure that you may see at some point in time, and every little dot, all the dots are intermediates. These are the carbon intermediates. So things like pyruvate, glucose, acyl-CoA, acetyl-CoA. And all the lines are the enzymes that catalyze the reactions. So you can see that this one looks pretty nice because they've color-coded it. And you can what I like about this is that, let's look at carbohydrate metabolism, which starts up there. And I'm guessing a certain number of these steps right here are glycolysis, but I know for sure that one right there is acetyl-CoA, because this circle is the TCA cycle. And what you'll notice is that almost all of the roads lead to the TCA cycle. So lipid metabolism over here will lead, sorry, I'm just trying to find the right connection. This one, where are you coming in from? This one will come in, lipid metabolism will go to acetyl-CoA because it has to eventually go to the TCA cycle. Amino acid metabolism enters into the TCA cycle. Nucleotide metabolism kind of connects into the TCA cycle, but the reason that everything connects into the TCA here is because TCA is the place where CO2 comes out, a lot of CO2, so it's the final ending point for catabolism. So you can see that we've actually done a good number of these. So we've done carbohydrates, we've done lipids, we've actually done a couple amino acids. I don't know if you've noticed. We've talked about some of our vitamins and cofactors, or what I would call coenzymes. Biochem 2 will do some nucleotide metabolism. We've done some of this. We've done some glycan biosynthesis and metabolism. We might get to xenobiotics, but anyways, I just think these images are really cool. It's a map of all the metabolic pathways, and again, the dots are the intermediates, what we might call substrates and products, and then the lines that connect the dots are the enzymes that catalyze those reactions. Okay, so you can't have both gluconeogenesis and glycolysis running at the same time, just like we can't have fatty acid synthase and beta oxidation running at the same time. These are futile cycles. So the focus is going to be, how is it that these pathways are controlled? So how do we get it so one path, so they're not both quote unquote on at the exact same time? And this is going to be all about how you can regulate enzyme activity. So again, this is the order in which we're going. We're going to start and just like truck our way down. So these are all the different ways that you can affect enzyme activity, and the enzymes are the ways that the pathways are controlled. So if you can slow down phosphofructokinase, or PFK, then you slow down glycolysis. If you can speed up fructose 1,6-bisphosphatase, then you speed up gluconeogenesis. Anyways, there's a number of different ways to do the slow down or speed up process. Okay, so the first two that we're going to talk about are pH and temperature. These are probably ones that you've seen before, but I'm going to combine them into one slide because I think this will actually make quite a bit of sense to you. So if you think about an enzyme, which is a protein, and I'm going to do here's an alpha helix, which is connected to another alpha helix, and then the protein, squiggles around and goes on and on and on. So what holds together the protein is the tertiary structure, right? And tertiary structure is an interaction between amino acid side chains. So in this case, I'm going to do an aspartic acid, and then over here, this will be a lysine. I'm not going to draw out the whole thing just because of space. So in this, a lysine and a glutamic acid, or I'm sorry, an aspartic acid, are forming a salt bridge together. Okay, and that's because of the pKa's and the pH's. Now, let's say that this pH right here is 7. At pH 7, this tertiary structure is optimally stabilized because the interactions are highly favorable. Now, if we move the pH to, let's say, 9, no, not that one. Let's do, why did I pick 9? I'm not going to move either one of them. If you pick 9, let's move it to, I gotta get back to my colors here. Let's go to 13. If we move the pH to 13, then this amine group becomes neutral charge, and we get down to, instead of a salt bridge, this becomes an ion dipole, which is weaker. So enzymes, and this image gets pixelated when I move into it too far. So enzymes have, and enzymes and proteins, doesn't have to be an enzyme, but a protein have optimal pHs, where the tertiary, and if it has quaternary structure, are ideally, not ideally is not the right word there, are stabilized, where the structures are stabilized. I could say where the structures are optimally stabilized. And this image right here is really nice because it shows 3 different enzymes, and they all have optimal pHs. So over here on the y-axis, this is enzyme activity. So the greater the number, the better the activity. And down here is pH. So at the peaks here, that's optimal enzyme activity. And you can see it drops off in either direction. If you lower the pH or you increase the pH, it alters the protonation state of the amino acids, which weakens the protein, and then it begins to unfold. So basically, at optimal pHs, the protein is folded, and at these pHs down here, it becomes unfolded. And when a protein is unfolded, it has no activity, or it has very little activity. So this pepsin is part of the stomach, so it makes sense that its optimal pH is somewhere down in the 2 to 3 range, because that's what the pH of your stomach is. Amylase, which is an enzyme that breaks down amylose, is in your saliva, and that pH in your mouth is about 7. It's not very acidic in your mouth. And this arginase right here in the liver, I mean, I can't tell exactly how far this goes, but let's say that's about 9 or 10. So each enzyme is folded at an optimal pH to optimize the non-covalent interactions between the amino acid side chains. And anything beyond that, it gets unfolded and the activity decreases. So temperature is actually very similar. You notice the curves look really, really similar. In this case, again, we have enzyme activity on the left-hand side on the y-axis, and then temperature down here on the x-axis. And it also makes a bell curve because there's also an optimum temperature. And the reason for this is not too hard to imagine. has to do with stabilization of the protein structure. So again, I will zoom. What colors was I using? It doesn't really matter what color I use. Let's put my aspartic acid back in, and part of the side chain of a lysine, and again we make a salt bridge. Now if you increase the temperature, you actually input enough energy so that at high temperatures all the non-covalents will break, and the enzyme, because we're talking about enzymes, it could just be a protein, unfolds. And remember, what I mean by unfolding is it goes from its tertiary or quaternary structure, depending, just down to its primary sequence, and the primary sequence doesn't have any enzyme activity because it's the 3D fold that gives the enzyme its function. So if you go too high in temperature, all the non-covalents will break. If you go too low, what happens is that everything just slows down, like really, really, really far, and the molecules, they don't vibrate as frequently, they don't crash into each other as frequently. So it's not really an unfolding that happens at low temperature, it's just that everything slows down. Okay, so that's how pH and temperature affect enzyme activity. This is not a way that pathways like glycolysis will not do this. Not a way that pathways regulate themselves. Because you only see high temperatures in your body when you have a fever and it's trying to unfold the proteins of the virus or the bacteria so that dies, but it turns out it also unfolds your own proteins. And again, if you alter the pH, you'll unfold all the proteins, and that would not be to your advantage as a human. Anyways, but I did want to bring these up because people talk about them all the time. High temperature unfolds a protein, low temperature just slows it way down, and then you really don't adjust the pH too much unless you have ketoacidosis or too much lactic acid buildup. But I did want to bring them up so that you had a little review from before, and I thought that maybe you would think, oh yeah, I do get that. Okay, so now we're going to move on to things that can actually change in the body. So there's a really nice graph that is, again, enzyme activity enzyme activity here, or maybe we'll call it reaction velocity or enzyme velocity. And then concentration of the enzyme here. We've seen this where this is S, we've seen it where this is L, and this is the first time you're seeing it where it's E. And it turns out the more enzyme that you have is a linear relationship to the activity. So if you increase enzyme concentration in the cell, you should increase reaction velocity. If you decrease enzyme concentration, there's a direct relationship where it will decrease. So if you want to turn down glycolysis, one way to do it would be to decrease the concentration of PFK, right? And then to turn up gluconeogenesis would be to, so this should be a concentration, would be to increase the concentration of F16BP, we call this TACE. Okay, that would regulate the output of the pathways. And there's a few ways that we can do this. We can use, there's something called Zymogens. There are ways in your body that regulate the half-life of protein, and those are through Pest sequences and the Nterminal sequence identity. And then there's transcriptional regulation, which is regulation at the DNA level. Okay, so Zymogens are inactive precursors, inactive enzyme precursors. And the way that you get these to be active is that they must be cleaved to form their tertiary structure and then have their activity. So in this case, the serine proteases all start out as zymogens. And so in this case, we have chymotrypsinogen. So it just plopped the end of the zymogen word onto chymotrypsin. So you could have chymotrypsinogen, elastasogen, and what's it, trypsinogen. And chymotrypsin is, and this is its primary sequence, from 1 to 245 residues. And the first thing that happens is a trypsin cuts right here between an arginine and an isoleucine, which makes sense because trypsin cleaves to the C-terminal side of positively charged residues. And then you get what's called pi chymotrypsin, and it has a little bit of activity and actually eats itself or autolyses for the second cut. And it cuts, remember, chymotrypsin is selective for large aromatic amino acids, so here it cuts after that tyrosine. And so you get your next cut there, and what ends up happening is you get 3 chunks. Oh, and there's a cut right here. So you get 3 chunks, chain A, chain B, and chain C. So again, we started with an inactive enzyme precursor. It's gotten cut 2 times. And then the way that it gets pieced back together is through disulfide bonds. So here are the disulfide bonds that cleave, or not cleave this, that piece this enzyme back together so that it has a 3-dimensional fold. And what's shown in red here is the catalytic triad. Okay, and if you didn't have this cutting and pasting, you wouldn't have the catalytic triad in the right orientation and proximity for catalysis. So again, zymogens are inactive enzyme precursors that must be cleaved into their tertiary structure in order to be active. All right, N-terminal sequence identity. I think this is really interesting and it's fairly simple. So N-terminal sequence identity is the amino acid that is right after your N-terminal methionine. So this would technically be residue number 2 in the chain. So if you remember back from your transcription and translation always starts with an N-terminal methionine. Well, it turns out that methionine actually kind of gets cut off, and a lot of protein sequences is post-translationally cleaved off, and then you're left with a different N-terminal residue. So that N-terminal methionine oftentimes gets cleaved off post-translationally, so after the ribosome works, and you're left with a new N-terminal residue, most N-terminal residue. So this would now be residue number 1. And this N-terminal residue is actually a marker for half-life. So if your N-terminal residue is an arginine, so that means an arginine is starting the chain, then that protein is likely only going to last 2 minutes in the cell. And it's going to be marked for degradation by the proteasome within 2 minutes. That's not very long. Proteins that I worked with when I was in graduate school had a proline as the N-terminal residue, and again, these also had really short half-lives in the cell, 7 minutes. And then there's some proteins that last more than 20 hours, but I think hemoglobin is one of the longest lasting, it's not targeted for degradation, and I think it lasts up to a week. But this just tells you that proteins don't last for forever. They get chewed up and recycled and rebuilt constantly all the time. Your cell is very, very dynamic. It doesn't make something and that lasts for forever. So things are being turned over all the time. So the N-terminal amino acid identity can determine how long the protein lasts, and that is directly correlated to the enzyme concentration. So if your protein has an arginine as its N-terminal residue, the concentration of this enzyme is always going to be really, really low in the cell because it's always getting targeted for degradation. Okay, the next one are Pest sequences. So these are amino acid sequences, so primary sequences that are rich in proline, that's the P, glutamic acid, that's the E, serine, and threonine. And proteins that are rich in these amino acids tend to last less than 2 hours in the cell. Weird, huh? Okay, so we've gotten through all of these. I'll actually come back to transcriptional regulation at the very end because I have a figure that pulls all of this together. And now we're going to do the rest of these. So the rest of these do not alter enzyme concentration. Rather, they alter the activity only. So they might change the shape of the enzyme so that it either becomes better at doing its reaction or worse at doing its reaction. And phosphorylation is a way of regulating enzymes. Phosphorylation, where you add on a phosphate group, is catalyzed by kinases. This is just vocab. So a kinase will add a phosphate to a protein, and dephosphorylation is catalyzed by phosphatase. And then actually sometimes the dephosphorylation can also be uncatalyzed. So in this direction, taking off the phosphate is catalyzed by a phosphatase. And typically proteins that are phosphorylated, they're phosphorylated on the hydroxyl groups of serine, threonine, and tyrosine because these are the amino acids that have OHs on them. And here's a diagram of what it looks like when a tyrosine is phosphorylated, a serine is phosphorylated, or a threonine is phosphorylated. That OH is a good nucleophile on a serine and can readily take off a phosphate group. I'm not going to draw all of them out. Can grab a phosphate off of, in this case it looks like I've got, I don't know, an ATP here and become phosphorylated. Phosphorylation is sort of like an on-off switch. So in this figure, the purple here is the protein, and this is a non-phosphorylated version, and the site of phosphorylation is right here. Now a kinase can add the phosphate group onto it, and what this does in this case is it causes the protein conformation to change. So that big phosphate group in this case is attracted to these positive charges, probably some arginines or some lysines, and it completely changes the shape of the protein, and the change in shape can either be an active form or a deactivated form, and it's actually impossible to tell in this cartoon diagram whether this activates the protein or deactivates it, but that giant negative phosphate group can change the shape of the protein to make it either more active or less active. And that's that one. All right, some of these we've already seen before, so kind of brace yourself for a little bit of a review on how substrates and inhibitors can affect enzyme activity. All right, so the reaction rate depends on the concentration of substrates. And if, how do I want to say this? The reaction, whoa, hey man, what happened? Reaction rate is very sensitive. at low substrate concentrations. Okay, so this is where the frequency of a substrate meeting the enzyme really matters. So if you look at this diagram over here, which is a Michaelis-Menten plot, so you have initial velocity here, you have substrate concentration here, and I don't know what enzyme this is, but I don't know, let's say it's hexokinase. Remember, hexokinase catalyzes the reaction of ATP plus glucose goes to ADP and G6P. So in this case, ATP is a substrate for this reaction. So what you'll notice is that at low substrate concentrations, that's this region right here, at low substrate concentrations, the rate is, I don't know, highly variable. It's highly influenced by even small fluctuations in the concentration of substrate. Whereas at high substrate, the enzyme is saturated and the rate doesn't change much. I'll do delta for change much. That's this region right here. So at high substrate concentrations, the enzyme is completely saturated up with substrate and the rate is unlikely to change. So what does this mean for regulation? What this means is that in a cell, the concentration of substrates, the concentration of products, what we've been calling carbon intermediates, is always kept low. This allows for even small fluctuations in a substrate concentration to have a big effect on the enzyme activity. So if you keep the concentration of glucose really low and it shifts up just a little bit, that's going to increase the reaction rate for hexokinase. Okay, and if it comes down a little bit, it's going to decrease the reaction rate for hexokinase. Okay, and on to inhibitors. So the inhibitor slides I'm going to kind of go through really quickly because you've already seen them before: competitive, uncompetitive, and mixed. Remember that competitive inhibitors bind to the active site and they compete with the substrate, which means that they look very, very similar to each other. In this case, the Km will decrease and the Vmax will stay the same. When you look at a Lineweaver-Burke plot, Vmax is staying the same. Km is altered in the presence of the inhibitor. And the reason that the Vmax stays the same is because at high concentrations of S, you're able to overcome the inhibition when all of E is bound to ES. And then the other two flavors of inhibition that we had met were mixed and uncompetitive. And both of these are types of allosteric regulation. And allosteric inhibitors, which these are both allosteric inhibitors, bind somewhere other than the active site, bind to what's called an allosteric site. Allosteric sites are just not active sites. You can tell this is an allosteric site. The active site is here. They are two physically separate locations. An allosteric inhibitor, whether it be mixed or uncompetitive, in some way prohibits either the enzyme from doing its reaction or the substrate from binding, so that makes it worse. There are also allosteric activators, where the binding of the activator actually makes it easier or better for the substrate to bind. So one example of allosteric activator that we've seen before is O2 binding to hemoglobin. When one O2 binds, it increases the affinity for the other O2s to bind. Food for thought. And again, just as a recap, uncompetitive inhibition is an order of operations. You must form ES, and then you form ESI, and the overall effect is Km and Vmax decrease in the presence of the inhibitor. Whereas mixed inhibition, the order of operations doesn't really matter. Either substrate binds first and then inhibitor binds to get to ESI, or inhibitor binds and then substrate binds to get to ESI, and what you get is a population average of what's going on, and the effect is that, oops, I was always doing Km first. Km increases and Vmax decreases, as you can see on the Lineweaver-Burke plot, which is this one right here. All right, I think I only have one extra kind of inhibition, and again, it's kind of a simple, straightforward one, and that's called feedback inhibition. So in many cases, the final products of a metabolic pathway directly or indirectly inhibit their own biosynthesis. So in this case, here's a metabolic pathway. It has 1, 2, 3, 4, 5 enzymes, and 1, 2, 3, 4, 5, 6 intermediates or substrates. We start with threonine and we end with an isoleucine. And in cases where you've been running the pathway for a really long time, you get a high concentration of isoleucine, that's a signal to shut the pathway off. You've made enough isoleucine, time to be done, so isoleucine will feedback, inhibit this first enzyme, and I actually have a cartoon diagram of what's going on here. So here we start. I don't know what this is going to look like when I zoom in. It's probably going to pixelate out pretty bad. So here's our threonine binding to the enzyme. And again, many steps, and you get to isoleucine. When you have high concentrations of isoleucine, isoleucine can feedback, inhibit this very first enzyme, which is called threonine deaminase, which probably means it takes the amine off of the threonine, the NH3 plus part, which is on the backbone. Anyway, so isoleucine can come up here. It binds to an allosteric site. Okay, it changes the shape so that no more threonine can bind, and then the isoleucine is used by the cell. So feedback inhibition is a really common way of inhibiting metabolic pathways, because the end of the pathway, the product of the end of the pathway, or a major carbon intermediate at the end, when it builds up, can be a signal that the pathway's been running for way too long and needs to be shut down. And is that it? No, that's right. I have this one diagram. I found a diagram that has everything in it. So what we don't talk about are extracellular signals. These things could be insulin, glucagon. Oops, that was supposed to be insulin, glucagon, etc. We will come to these if you end up ever being able to take Biochem 2. We will talk about hormone regulation. But let's do one at a time. So this one right here is the transcriptional regulation. And so if you use a transcription factor to either upregulate or downregulate transcription, that's this arrow, translation is here, what you get, transcriptional regulation affects the concentration of enzyme. So if you have a transcription factor that upregulates, then you get more enzyme. If you have a transcription that downregulates, you get less enzyme. And again, remember that enzyme concentration is directly related to activity. Okay, so what else? Phosphorylation is right here. And you cannot predict whether adding a phosphate or taking it off is going to be, it's not like always adding a phosphate makes it active and always taking it off makes it deactive. Those are things that work together. It's completely dependent on the enzyme. Right here, the protein degradation, that one is your Pest sequences and your Nterminal sequence identity. These right here are how your concentration of S affects enzyme activity, and all of your concentrations of inhibitors affect enzyme activity. And I think that's just about every one of them. We won't really talk about regulatory proteins and how they inactivate or deactivate. Okay, so there's a whole list of things that you'll need to kind of keep straight, but we're going to apply some of these concepts to the metabolic pathway regulation, which is going to be part 2. Okay, sounds good. Bye-bye.